All right, where we left off, the last thing I'd been talking about was this technique called PCR. It makes it possible to copy any piece of DNA very accurately. And we can take a piece of DNA from, let's say, a crime scene, copy it very accurately, measure its length with a technique called gel electrophoresis. And that's how the simplest form of DNA fingerprinting is done. That's what we ended with. You can modify the technique just a little bit. I'm not going to go into exactly how, but it's possible to read the order of nucleotides base by base. And there is, when I was your age, PCR had only just been invented. Carrie Mullis got the idea in 1985, and I was a college freshman in 1987. So it was still very cutting edge. It's now routine, and not just human, the complete, well, not the complete, because we don't all have identical DNA, but there are probably thousands by now of humans who've had essentially their entire set of DNA sequenced nucleotide by nucleotide. And there are many thousands, maybe tens of thousands of species of microbe, animal, plant, fungus, protist that have had at least part of their DNA sequence read. And this is stored in a huge database called GenBank that's maintained by the National Institutes of Health. And the cool thing is that anybody can search this and can find DNA sequences from hundreds of thousands of species, probably 100,000 by now. I don't even know the number, but it's a lot. And we can use this data as a whole new line of evidence for reconstructing evolutionary history. You might remember we talked about evolution quite a bit. Well, just to give you a quick example, these are some short snips of DNA from a hippo up on the top and three different whales. The orca, aka the killer whale, although there's no documented cases of orcas ever attacking people. They do attack other things. They're pretty badass predators. Sperm whale and blue whale, the largest animal of all. And these are short snips of their DNA. And if you look at them carefully and you advance the slide, you can see that there are some differences nucleotide by nucleotide, not just between the hippo and the whales, but between different groupings of the whales. Let me see here. If you look right here, you can see that at position 5, where the hippo has C, all three of the whales have an A. And at this position, all three of the whales have an A, where the hippo has a G. And over here, all three of the whales have a C, where the hippo has an A. So that's 3 similarities that the whales all have in common with each other that's not present in the hippo. The simplest explanation, not the only, but the simplest, is that all 3 whales descend from a common ancestor that also had those 3 points of similarity. And then if you look, you can see that within the whales, over here at position 6, you have orca and sperm whales have an A, where the hippo has a G. And here at 7, orca and sperm has a C. And over here, orca and sperm has a C. So there's 3 points in which the orca and the sperm whale share a nucleotide that the others don't. The simplest explanation would be that the orca and the sperm whale share a common ancestor that wasn't shared by the others. Oh, okay. I am told cases of orcas attacking people are all in captivity. In the wild, they don't, or at least it has not been reliably recorded. Okay, thank you for setting me straight on that. All right. There's some other bits and bobs and things like that, and all of these changes result from mutation. And we can take, when these days we would not be doing this by eye, but we can take sequence data like this, plug it into computer software for various types of analysis, and we can draw up these diagrams that you might well remember called cladograms that we talked about way back in around about week 5. And on the basis of this, it's currently thought that toothed whales, like sperm whales and orcas, share a common ancestor, and that ancestor shared an ancestor with baleen whales, which are whales that don't have teeth, but have a special filter apparatus in the mouth, like blue whales and also humpbacks and right whales and things like that. So all whales, we think, come from a common ancestor represented by that spot. And that common ancestor had an even more distant common ancestor with hippos. Hippos are the closest land relatives to whales. And then that ancestor shared an even farther back common ancestor with the ancestors of little deer-like mammals called chevrotains, and a big group called the pecorans, which is giraffes and cows and goats and sheep and deer and antelope and moose and Chinese muntjacs and so on. And so remember, I talked way back when about how we can use similarities to draw up hypotheses of shared common ancestors in evolution. Well, now it can be revealed that we can use a great deal of data that's available in DNA sequences to draw up hypotheses of common ancestry. And there's various ways we can test those, and we can keep refining them. For example, we can cross-check this against the fossil record. And we do know that the very earliest whales had sizable legs and do in fact resemble, they were land mammals that do have some similarities in common with hoofed mammals like this. So the fossils are pretty consistent with this picture, and we can use each one to check hypotheses derived in the other, right? We test hypotheses. It's what we do. We also use this to track the evolution of virus strains. So this very branch diagram right there, these names right there that are written in red, that is your friend and mine, COVID. Those are strains that were isolated in China back in 2019. They share a common ancestor with other coronaviruses that have been isolated from bats. All of these right here, those are mostly bat coronaviruses. They share a more distant common ancestor back here with some viruses that include the virus for MERS. That's Middle East Respiratory Syndrome. That caused a severe outbreak of disease in the Middle East. I believe it's more dangerous than COVID is, but fortunately it didn't go global. It never became a global pandemic, but it was pretty big news in, I want to say 2005. I think it was about 15 years ago that MERS was a big deal. And then we can trace common ancestry with yet other viruses back here from minks and ferrets and mice and also bats, mostly bats. So we can track the evolution of coronavirus, and we can find new mutations of it, new variants of it, and we can work out where they come from, and we can work out how they evolve. And that's what you can do when you have PCR and you get the sequence of a lot of DNA, and then you use basic evolutionary reasoning that we went over many weeks ago. You can do things like this. And we can push it back and we can explore the evolution of all life on Earth. And it turns out that even bacteria and archaea, the living things that are probably least familiar to you and least like you, turn out to share some very deep genetic similarities with you. You're over like here somewhere. And all other animals and fungi and this thing right here that says Archaeplastida, that's plants and algae, and these are various groups of protists, and all the way to bacteria and archaea. As far as we can tell, life on Earth only arose once, and all life forms are related if you could go back far enough. And a sizable part of the evidence for that has come from DNA sequencing data. Okay, again, this is not going to be on the second exam. You are going to see it on the final in about a month. Okay, so let's look at a little bit more at some of those deep similarities I talked about and look at the structure of cells. This is something we've kind of fancy-footed around, but I haven't really gotten in detail about it. These are a couple of protists. They both happen to be, well, of course, they're eukaryotes because they're protists. And if you were curious, the nucleus of this one I'm shading in blue. And the nucleus of this one is more or less this bit around here. And any living cell, prokaryote or eukaryote, contains a liquid substance inside called cytoplasm. And cytoplasm turns out to be a very complex solution of mostly water, but there's various salts dissolved in it. A lot of proteins may be floating free. Various other substances may be floating around in the cytoplasm. And everything inside the cell floats in the cytoplasm or is held up in the cytoplasm, is suspended in the cytoplasm. You can see lots of little bibs and bops and deedly bobs inside this amoeba. We don't really need to worry about what those are, except suffice it to say that they are mostly floating inside that cytoplasm. And the same is true for the little green dots in this Paramecium bursaria protist right over here. They're all pretty much floating in the cytoplasm, suspended in it. What surrounds the cytoplasm is a membrane. It's called the cell membrane, or you'll also see it called the plasma membrane. And you might remember, and in fact, I think you're doing this in lab this week, where you're looking at the chemical components of various foods. There's a major class of macromolecules called lipids. And we talked about lipids way back at the beginning, that all life has these 4 classes of big molecule, proteins, nucleic acids, carbohydrates, and lipids. Well, lipids include fats and oils. It also includes some things like waxes. Your earwax is a lipid, so is beeswax. But there's other types of lipids, and if you add them to water, they tend to bunch together in a particular way and form sheets. And your plasma membrane is basically made of lipids forming a sheet. And you can get proteins and other molecules that might be embedded in the membrane and can stick out of one side or stick out of the other side. And we don't need to worry for our purposes about what those do. Some of those allow the cells to transport molecules in or out. Some of them allow the cell to sense signals from the outside and respond to them in various ways. Some of them enable a cell to bind to the extracellular matrix if there is one. Again, we don't need to worry about what all of them do. Let's just say that what keeps the cytoplasm in is this plasma membrane made up of a particular type of lipids. If you must know, they're called phospholipids, and they form a structure called a phospholipid bilayer. Don't worry about all these names of different types of proteins like integral proteins, peripheral proteins, and alpha helix proteins. Don't worry about that bit. For our purposes, it's not critical. And one thing to keep in mind is the membrane isn't really a solid. The membrane is technically a very thin film of liquid. If you've ever looked really carefully at soap bubbles, if you're a kid, you're blowing soap bubbles like this, you might be able to see that, on the surface of the soap bubble, you can see, little bits of foam flowing over the soap bubble. You might be able to see that soap film kind of swirling. So the soap that makes up a bubble is still a liquid substance. It just blowing the bubble makes it form this nearly two-dimensional sheet that wraps up into a bubble, wraps up into a sphere. This is a pretty good metaphor for the way that cell membranes work. They're not truly solids, not like a sheet of rubber or something like that. They're technically very thin liquid films. And because cell membranes are liquid films, it's very easy for them to either fuse or pinch apart. If you've ever really, I don't know, I sound kind of like some of the druggies I used to know in Berkeley. If you ever really looked at a bubble, man, like especially on shrooms, dude, whoa. Okay, try not to sound that way. But if you've ever really looked at like your dish soap or, or foam in your sink or something like that, when you wash dishes or in your bathtub, if you take a bubble bath, if you watch that foam carefully, you'll notice that these bubbles sometimes join together. There'll be two small bubbles next to each other, and then the membrane between them pops, and all of a sudden you've got a bigger bubble. Or you might have a big bubble, and you find that if you poke it just right, and you may not get it to pop, but you can split it into smaller bubbles. This is basically how cells work, and this is how cells can divide. Cells can also grow in size. It is beneath a sophisticated, sorry, I've got a comment. It is beneath a sophisticated professor to act like a hipster. Okay, well, if you ever take a class from a sophisticated professor, you can tell them that. Yeah, this is a fairly good model for how cells work. This is what makes it possible for cells to divide. When a cell divides, you're not having to tear anything the way you would a rubber balloon or something like that. You're simply taking that two-dimensional film of lipids and pinching it off in much the same way you could do with a bubble. And you see this, and yeah, you see this a lot inside the cell. There's a lot of structures made of membrane inside the cell, and they're constantly fusing together and pinching apart. And I'll show you some of that in just a bit. Prokaryotes, as I hope you remember, most of them have a cell wall outside the cell membrane. There's actually a few that don't, mostly disease-causing bacteria, The bacterium that causes the disease, typhus, is one. But most bacteria do have an external cell wall. And again, that's where it is. And the cytoplasm here in this particular picture just looks like just grainy stuff. But inside there's kind of a central area where there's a lump of DNA, and it's called the nucleoid. And that's that lighter colored area that looks kind of like a bunch of spaghetti. But this isn't bounded by a sac made of membrane, and so we don't call it a nucleus. It's just a heap of DNA, and ribosomes are just kind of floating freely in the cytoplasm. If you look outside the nucleoid, all these little black dots, most of those are probably ribosomes. And now it can be revealed some more of the differences between prokaryotes and eukaryotes. Prokaryotes, bacteria, and also archaea, the DNA is organized into one big loop. Your DNA, like all eukaryotes, is arranged in strips, and every strip is a chromosome. A chromosome is just one long piece of DNA, you know, that like any piece of string, it's got a beginning and an end. Bacterial DNA, you'll typically get one big loop and then a number of small loops called plasmids. And this is a couple of plasmids seen using a type of electron microscope. The magnification is still not good enough to see the actual double helix, but it is there. These are pieces of DNA, and you can see they look like rubber bands, right? They're loops. And bacteria swap plasmids the way kids swap Pokemon cards. They can exchange plasmids with each other. In fact, when bacteria have sex, what they do is build a bridge between them, and one of them sends a copy of a plasmid over to the other. Bacterial sex is basically a form of file sharing. Kind of a weird way to think about it, but there you go. Bacteria can also pick plasmids up from the environment, and that's what was happening when Griffith did his famous experiment showing that living bacteria could be transformed by picking up something from dead bacteria. What that something was DNA plasmids, circular pieces of DNA that the dead bacteria had inside them that got released when the bacteria died and broke open. Now it can be revealed. Hopefully you still remember that eukaryotes have a nucleus. It seems I have misspelled it nucleus on this slide, but that's supposed to be a nucleus. And what surrounds the nucleus is a double-layered nuclear membrane, and it's made of the same lipids as the outer membrane of the cell, the plasma membrane. All membranes in a cell are made in basically the same way. They're basically these films of specialized lipids. If you look carefully at nuclei, there's often a darker spot inside the nucleus called the nucleolus. And what you're looking at here, this happens to be a plant cell, but your cells aren't all that different. This right here that I'm tracing in red, that is the nuclear membrane. The grainy stuff inside, that's all DNA, and it is separated into chromosomes. But remember that when a cell is not actively dividing, when it's in interphase, you can't see the chromosomes because they are unwound and jumbled up together. So that's why you can't see individual chromosomes here. You just see this kind of gray goo. But there's that darker spot inside, that is called the nucleolus, and I'm highlighting it in blue right there. That happens to be the spot where ribosomes themselves get made. There may be more than one nucleolus. It depends on the species and the type of cell you're looking at. So that's the ribosome factory, the nucleolus. And to allow ribosomes to get out of the nucleus and go to where they're needed, and to allow to get things like messenger RNA to leave the nucleus and go to where it needed, the nucleus, the nuclear membrane has gateways in it called nuclear pores. What you're looking at is a cell that was flash frozen and then hit with a knife to break, and then imaged under a scanning electron microscope. And what you're seeing is the surface of the nucleus, and you might notice that on that surface you have these little dots, which I guess I'm kind of covering up by highlighting them right here, but you can see there's quite a lot of them. This thing looks kind of like a pepperoni pizza. What those dots are the nuclear pores, and there's actually proteins that form a gate-like structure that can open and close. So the nuclear pores can open to let messenger RNA out, and then close behind it. So that's how mRNAs and certain proteins get into the. That's how proteins get into the nucleus, and messenger RNAs get out of the nucleus. Okay. Inside a eukaryote, there are a lot of structures that are made of membrane, and they're made of the same type of lipid bilayer membrane that the plasma membrane on the outside is made of. And probably the most important part is a transport network within the cell that is made up of lots and lots hang on, sorry, I got somebody tried to join and I think I lost the, hope I admitted the guy. If not, I hope they'll try again later. Right. Okay. This transport network consists of lots and lots of membrane folded up into folds and tubes, and we call it the endoplasmic reticulum. Reticulum is Latin for a net, a little net. So this is the little net inside the cell, and endo is inside, and plasmic has to do with cytoplasm. So this is the little net inside the cytoplasm. If you don't feel like calling it the endoplasmic reticulum, you can just call it the ER. And proteins and other products made inside the cell, especially ones that are exported out of the cell, move via the ER. So if you think for a minute of a cell as being like a protein factory, the ER is the factory floor. The ER is connected to a body called the Golgi body. If anybody's curious, Camillo Golgi won the Nobel Prize back in 1905, and he's the guy who discovered this. He was an Italian biologist. And the Golgi you can think of as the packaging and shipping center of the factory. Any proteins that have to leave the cell tend to go by way of the Golgi, and they tend to get sorted there. And then on top of all of this, you can have sacs of membrane in the cell that are called vesicles if they're tiny, and vacuoles if they're large. And they can be used for storage or for transport. So that pictures a slice through a eukaryotic cell, magnified maybe 100,000 times, using a gizmo called a transmission electron microscope. And what you're looking at is a cross section through the ER. You see, it looks like a lot of parallel lines. What you're looking at is slices through membrane. So those parallel lines, you're actually looking at a bunch of flat membrane folds and sacs, but you're looking at it like you're looking at it in cross section. And if you look carefully, in some of these folds and sacs, you can actually see little dots, little black dots. This type of ER is called rough ER because those black dots make it look rough, like you're looking at, sheets of sandpaper or something like that. And those black dots are actually ribosomes. So a cell that makes a lot of protein to be exported is going to have a lot of rough ER. I mentioned this a while back, but you've got cells in your salivary glands. You've got two kind of at the angle of your jaw and one up under your chin. And those are the glands that produce saliva and pump it out into your mouth. That's why your mouth is nice and moist inside. And your saliva is not just water, it contained enzymes that break down starch. I know we mentioned this a long time ago. The cells in your salivary glands would probably have lots of rough ER because those cells make the enzyme alpha amylase. And then that alpha amylase is exported out of the cell and it flows with the rest of your saliva into your mouth. So cells like the cells in your salivary glands make lots of protein and then push it out of the cell. And cells that do that have lots of rough ER. You can also have smooth ER, which looks like this in cross section. You're looking at a big network of membrane tubes that you've sliced through. And even though there aren't ribosomes in the smooth ER, there can be lots of enzymes. So for example, your liver cells have lots of smooth ER because they contain a lot of enzymes that detoxify various poisons. Remember we talked about alcohol, which your liver has to break down to first acetaldehyde and then to acetic acid. And if we block that process from happening, you get the hangover from hell before you've even got a buzz. Well, that all takes place in liver cells. And the enzymes that are doing it in your liver would be located in the smooth ER. Cells that produce fats or carbohydrates would also be expected to have lots of smooth ER. So in the rough ER, that's rough because it has tons of ribosomes. And the ribosomes do what ribosomes do. They carry out translation and they make proteins, and the proteins pass through all of those folds and tubes in the ER, and they end up in the Golgi body, which looks like this. It's basically a stack of flat membrane sacs. Like imagine you had 10 deflated beach balls and you stack them one on top of the other. That would be a decent model of the Golgi. And what you're looking at here is a slice through that stack. So all of this right there that I've just shaded in blue, that's your Golgi. And you might notice that there's a whole lot of little bubbles up here. Like there's one. I'm not going to circle them all, but you have lots of little bubbles right here. Those bubbles are called vesicles. And there's some surrounding it right here, some surrounding it right there. There's one. And the point of vesicles is remember that living membranes are kind of like soap films and they can bud off or they can fuse. The Golgi is constantly budding off little vesicles. and having other vesicles join onto it. So there's always vesicles budding off the Golgi and always vesicles coming in and fusing with the Golgi. And so anything that the cell secretes goes into the Golgi. It might get modified in various ways. These vesicles bud off, and the vesicles from there can move to the outer membrane of the cell and can fuse with it to release the contents. Now, that's a process called exocytosis. Do not worry about the terms in here like constitutive secretion versus regulated secretion. We don't need to get into that. This is just a diagram of what this process looks like, with vesicles budding off the Golgi, moving through the cell, coming up to the cell's outer membrane and fusing with it, releasing whatever is inside them to go out into the big wide world. And this is going on all the time. And it's not shown here, but they can be vesicles that will fuse with the Golgi as well as leave the Golgi. Okay, so we talked about vesicles. Those are small. Some cells have very large bubbles in them, and when they're big like this, we call them vacuoles. You're looking at a cross-section through plant cells, and you'll notice that like this whole thing that I'm outlining in blue is one big plant cell. And yes, it has got cell walls on the outside. And a lot of the space inside that plant cell is empty. That's the vacuole. Vacuoles in plant cells contain mostly water. And even though they don't contain much else in them, they're just big bags of fluid, they're important in structural support. If you take a leaf of lettuce that's freshly picked, it's going to be nice and crisp. If you leave it out and you don't keep it wet, as it dries out, it's going to get really floppy. That's because as it dries out, it's losing water pressure inside that central vacuole. When that central vacuole is full, it's very firm. You know, just like a water balloon that's full of water is pretty firm. If that vacuole starts losing water, it stops being firm and eventually becomes flabby, like an underinflated water balloon. And that's basically what wilting is. When your lettuce or your spinach goes from being nice and crisp to being flabby, that's because they've lost water out of that big central vacuole. All the cells have lost water out of their central vacuoles. This is why, you know, if you go to the supermarket produce section, the leafy vegetables are periodically sprayed with water from this little, you know, misting system. It's to keep the the stuff from wilting as long as possible. Give you nice, crisp, fresh lettuce. Okay, what else is there? This is a cross-section of something called a mitochondrion, this thing that looks kind of like a sausage. And evidently this is funny because it's some kind of meme, and I'm supposed to tell you that mitochondria are the power plant of the cell. This is apparently an example of the kind of useless knowledge that you learn in school that has nothing to do with the real world. Well, your mitochondria are basically keeping you alive because most of the work of converting the food you eat into usable chemical energy takes place in the mitochondria. Quick example. You find lots of mitochondria in muscle, of course, but you especially find them in muscle that has to work for long periods of time. Muscle where you need endurance. Those muscles need lots of energy, and they've got lots of mitochondria, lots of these little power plants. Other types of muscle doesn't have a lot of mitochondria, and muscle like that can contract rapidly, but it gets tired very quickly. In people, the difference isn't immediately obvious. The difference between muscle cells with lots of mitochondria and muscle cells with few is the difference between what we call slow twitch and fast twitch muscle fibers. And every muscle in your body is kind of a mixture of those. How much you have of one versus the other may determine how naturally good you are at sprinting versus marathon running. But if you look at birds, for example, what the heck, let's look at chickens. Chicken flight muscles in the breast and in the wings can produce a lot of power. You know, they can go flap, 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 flap really quickly, but they get tired because chickens don't fly very far. Domesticated chickens may not be able to fly at all. Wild chickens can flap, flap, flap for a bit, but they don't make long migrations, right? You've never looked overhead and seen a majestic flock of chickens soaring overhead on their way to their summer breeding grounds 1,000 miles away. Chickens don't do that. What chickens do an awful lot of is walking around and scratching. And so the flight muscles in the breast don't contain a lot of mitochondria because they're optimized for speed, but not for endurance. The leg muscles do have to be optimized to endurance because a chicken is on its feet all day. And so one of the reasons why light meat and dark meat look different is that dark meat has lots of mitochondria and light meat doesn't have nearly as much. So you're eating a lot more mitochondria if you're eating a drumstick as opposed to eating the breast. So what do I mean when I say mitochondria are the power plant of the cell? I don't have time for all the details on this. Let me give you the Cliff Notes of the Cliff Notes. This is a compound called ATP, stands for adenosine triphosphate, and you might have noticed it is in fact a nucleotide. Right over here is the base, this, and it happens to be the base adenine, same as you would find in your DNA. And then this thing right here, that's the sugar, and it happens to be the sugar ribose. And then over here, that's a phosphate group. But look at this, here's a second phosphate group, and here's a third phosphate group. This thing has 3 phosphates instead of a single phosphate. But other than that, it would be perfectly usable in your DNA. What ATP is capable of doing, there's ATP, there is your 3 phosphate groups, 1, 2, 3. And what ATP can do is give up one of those phosphate groups and stick it onto another molecule. So right here, we've got, this happens to be the amino acid serine. What the ATP has done is given up one of its phosphates and stuck it onto the serine. And now the ATP is called ADP, adenosine diphosphate, because it's got 2 phosphates instead of 3. What the heck is the point with that? What that does is transfer energy to the to the serine. The serine not only has a phosphate group stuck to it, that serine has got energy added to it. And that means that the serine can now participate in a chemical reaction that it couldn't otherwise do. So when ATP gives up a phosphate group, it conveys some energy to the molecule that receives it, which means that ATP is ultimately the fuel that runs your metabolism. Simple example, here's somebody who is definitely flexing muscles very quickly. Your muscles are filled with proteins, and two in particular are called actin and myosin, and they're arranged in these long filaments that are side by side. Your muscles made of these protein filaments, which is in part why meat has a grain, right? If you've ever carved a turkey or a brisket or something like that, you know that meat is fibrous and you can cut it against the grain or with the grain. This is why. So you have these two proteins called actin and myosin, and they lie next to each other. And when ATP gives up a phosphate to a molecule of myosin, it causes the myosin to change shape. And when it changes shape, it slides past the actin. And then it happens again. And every time you burn an ATP, the myosin molecule goes ka-chunk and moves one space down the actin molecule. That happening millions of times over and over again is ultimately how your muscles contract. ATP gives up phosphates to allow your myosin molecules to change shape and go ka-chunk, 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 ka-chunk past the actin molecules, which you can think of for our purposes as holding still. And that's how your muscles get shorter. Myosin molecules going ka-chunk, 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 and every ka-chunk is the burning of one piece of ATP. So yeah, there's somebody with a lot of muscles that I'm sure is burning a lot of ATP. He seems to be feeling the burn. And your mitochondria carry out this beautiful chemistry that we just haven't got time to get into, that extracts energy from your food molecules and turn ADP back into ATP. In fact, every ATP molecule in your body loses a phosphate and then goes to the mitochondria and gets a phosphate stuck back on and turns back into ATP roughly between 500 and 750 times each day. If you turned ATP into ADP and couldn't turn it back, you'd burn your own body's weight in ATP each day. But that doesn't happen because it's your mitochondria that take energy from breaking down your food and use that to rebuild your stock of ATP. Oofta. Okay, so now you know why mitochondria are the power plants of the cell. All right, and why that matters just a little bit more than being the punchline in a meme. You wouldn't last 10 seconds if your mitochondria all shut down. The vast majority, not absolutely all of it, but the vast majority of the energy that your cells need to carry out metabolism and do absolutely everything ultimately comes from mitochondria. Okay, we've got a second type of organelle that's present in photosynthetic eukaryotes. They're called chloroplasts. Mitochondria are almost always too small to see under the light microscope, and even if you can, they just look like clear little dots. Chloroplasts tend to be bigger, and they tend to be green. And so I know you saw these when you looked at that water plant, Elodea, way back in lab number 2. There were these, you would have seen something like this, and every cell contains those little green discs that are floating around inside. Those are the organelles where photosynthesis takes place, which is just the way in which certain cells can harness solar energy in order to take things like carbon dioxide gas and water and turn them into larger molecules that the cell needs. This is how plants can live on CO2 gas, water, and a few minerals and a source of nitrogen for flavor. is that they're basically able to harness solar energy to run the building up of larger molecules. And then when you eat plants, you break those down and you get the energy for yourself. Okay. We'll be done pretty soon. I know this is a lot, but remember, you're not going to be tested on it this Tuesday. There's a network of proteins, and there are a couple of types, microtubules and microfilaments. Don't worry about the difference. Collectively, they make up what we call the cytoskeleton. You're looking at 4 cells here that have been stained with a type of fluorescent stain. That's why they look like they're glowing. The nucleus has been stained in yellow, and all of that red stuff that looks like cotton candy is that network of microtubules and microfilaments, the cytoskeleton. This enables the cell to keep its shape. For some types of cells, like those, all of those white blood cells that are a part of your immune system, they enable those cells to move. That's what allows your white cells to crawl through your body. Last thing, there are some eukaryotic cells that have these hair-like structures sticking out that beat back and forth. In some cases, these hair-like structures will push the cell through fluid. Many protists that can swim have these. So do sperm cells, at least of many animals. Not all animals have sperm cells that can swim. You do. Well, about half of you do. And they're able to swim because they've got these long hair-like structures sticking out that can beat back and forth, using up ATP to do it. And if they're long, those structures are called flagella. If they're short, like the ones that you're seeing here, those structures are called cilia. So typically a cell will either have one or a few long flagella or large numbers of short and stubby cilia. Cilia, by the way, is Latin for eyebrows. So just like your eyebrows are made up of many very short hairs, many short hairs is what cilia look like. You're looking at cilia right here. But the same basic structure is present in both. The only real difference is length. And not all eukaryotic cells are going to have these. Your sperm cells would have flagella. The cells inside your lungs are covered in cilia. So when you breathe in dust particles get trapped on mucus, and then the beating of the cilia pushes the mucus up and out of the lungs and eventually gets it to where it can be coughed up. That's how your lungs stay clear of dust. I mean, ultimately, if you inhale a lot of dust and overwhelm your cilia, you can end up with lung disease. Like if you breathe in a lot of coal dust because you're working in a coal mine, or for that matter, if you smoke, you can overwhelm your cilia. But this is at least how, this is at least what they're good for. Okay, yeah, I had that cold a couple of weeks ago, and yeah, my cilia were no doubt working overtime, pushing all that mucus out of my lungs. Okay. I tell you what, I'm going to save this until after the exam. Because what I want to talk about next is where all of that complexity of eukaryotes comes from. It might seem mind-bogglingly complicated, but there's a beautiful idea, a beautiful hypothesis that turns out to be very well supported that explains where so much of that complexity of eukaryotes comes from. And if you want to look ahead, it was worked out by a scientist named Lynn Margulis, who I actually had the pleasure of meeting about 20 odd years ago. She was a very nice but very strong person. And she published this in 1970, and we will get into it after the exam. So we'll look at it in 1 week. In the time that we've got, don't go away just yet. In the time that we've got, I want to show you a video, and I am going to stop the recording right now, but I'll take this video and I will post it to the playlist so you can go back and look at it. I just don't think I'm allowed to incorporate it in here. So I will now stop the recording.